Hello everyone. For those who don't know me, my name is Anwar Bayev. Glad to see you on my BioCulture channel and welcome to my SAT biology course. If you haven't subscribed yet, then be sure to be subscribed today, press like and share the channel with your friends and colleagues. Today we will consider the first type of macromolecule, carbohydrates. So before we begin, let's dwell on what macromolecules are. Macromolecules are large biological molecules that are made up of small particles. These include four main classes. We have carbohydrates, fats, proteins, and nucleic acids. Perhaps you ask, what do we need to know about macromolecules to pass the exam successfully? You must know certain terms and understand the general logic, the very essence how everything works. Let me list. First, as I said earlier, you should know that macromolecules are made up of small particles. You can imagine this in the form of bits on a thread folded into one product. So now carefully. The bit is our monomer, the thread is the bond between the monomers, and the product itself is a polymer, or in other words a macromolecule. Secondly, you must understand how bonds appear or become destroyed. To do this, pause the video and look at the diagrams on the left. In addition to all this, I would like to note that you should be familiar with the names of monomers and polymers, because with each class, they accordingly vary. So let's move on. Carbohydrates are our sugar, and glucose is a simple example. Here the monomer is called a monosaccharide. Its general formula is CnH2nOn, where n is any digit equal to 3 or more than 3. You may wonder, why is this formula not suitable for polymers? This is all because, as you remember, when forming bonds, water molecules come out, which leads to a decrease in the amount of hydrogen and oxygen in the formula. Here also you should know in what forms the monomers exist. They can be presented either in linear form or even in a ring-shaped form. The last of them manifests itself only in aqueous solutions, because this form is more stable under physiological conditions. Next, let's look at the sources of variety of carbohydrates. In general, there are three main sources of increasing diversity. First, this is the location of the carbonyl group. In other words, if you look at the diagram on the right, you will see that this group, CO, is either inside or at the end of the molecule. The second is the size of the carbon skeleton, or to put it another way, this is the amount of carbon in the composition of carbohydrates. Thus, using the Greek cancels, we subdivide them into trioses, tetroses, pentoses, hexoses, etc. Third, this is the spatial arrangement of the OH and H groups. As a result of all three sources, we get a huge number of different molecules, although their formulas may be the same. You can notice this in the same diagram on the right. I think everything is clear here. Let's move on. Disaccharides, so we call dimer, that is, two connected monomers. Here you in principle should only know which combination of monosaccharides lead to certain disaccharides. So first, maltose. It is formed by a combination of glucose plus glucose. The second is sucrose. It is formed by a combination of glucose plus fructose. And the last, lactose. It is formed by a combination of glucose plus galactose. Besides all this, you should not confuse the endings in the name of disaccharides and their enzymes that break them down into monosaccharides. For example, sucrose and sucrase. Next is variations in glucose structure. Glucose is, as we already understood, a monomer. So glucose can be of two types, alpha or beta glucose. The main difference is the location of OH group on the first carbon. If it is below, then it is alpha glucose, and if above, then it is beta-glucose. As a result of which, this leads to differences in the types of bonds between the two monomers. In other words, if we look at the diagram at the very bottom, we will see that if our polymer is made of beta-glucose, then beta-glucose monomers are located upside down relative to its neighbors. And as for alpha-glucose, when forming a connection, they do not change their location in space. Also, besides all of the above, you should know what effect they give in polymers. If the polysaccharide is made from alpha-glucose, then the polymer is more branched in the shape. And if it's made of beta-glucose, then it's more straightened than branched. So, let's move on to our last aspect of today's video, namely the polysaccharides. There are many types of polysaccharides. However, you need to know only 4 of them. The first type is starch. In this form, plants store glucose, or in other words, energy. The second type is glycogen. In this form, animals and we store energy in the body, namely in the muscles and in the liver. The third type is cellulose. It is present in the cell wall of plant cells. And the fourth type is chitin. It contains nitrogen and it is present in the exoskeleton of insects. Okay, on this our third lesson ended. As usual, I will leave a link at the description to download today's slide. I hope it was the most useful for you. And if you haven't watched my previous videos, be sure to check them out. I will be waiting for you in the next lesson. Всем большой рахмет!